This is episode 290, the Stem Cell Podcast, ISSCR 2025, Illuminating the Future with Drs. Valentina Greco, Kathryn Cheah, and Eugenia Piddini. Hello, everybody. We are Drs. Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. And we got them today, 3 of them. Excited about this chat, excited about the meeting coming up, traveling to Hong Kong. We're going to talk about all that and more. This podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. As I said, we got some hitters today on the show. Drs. Valentina Greco, who's also the president of the ISSCR, by the way. Kathryn Cheah, who has led the bid committee there, and Eugenia Piddini, who is on the program committee, but also esteemed researchers in their own right. They're going to talk about the upcoming ISSCR meeting 2025. That's happening June 11th to the 14th. And as I said, in Hong Kong, Arun and I are really thrilled. Looking forward to that one. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, and on the topic of ISSCR, just a reminder to submit your late-breaking abstract for the annual meeting. Submission closes on March 19th, so just around the corner here. Don't miss your chance to submit your abstract. You can learn more at isscr2025.org/abstracts. Something that's going to be talked about a lot at ISSCR, undoubtedly, are organoids and ways to improve them. So the first article I'm going to talk about here in the roundup for today is a cell stem cell article titled Coordinated Differentiation of Human Intestinal Organoids with Functional Enteric Neurons and Vasculature. I mean, we've been talking about recently ways to make these organoids better. And you got 2 things right there in the title. It's introducing additional tissue types in the differentiation, like enteric neurons, as well as improved vasculature. I think anybody who's working with organoids they're trying to approach these questions and answer some of these questions about how to make organoids better. And this is first author Charlie Childs, and this is work coming from the lab of Jason Spence over there at the University of Michigan. So we've talked about HIOs, human intestinal organoids, before on the show. I mean, that just recently as within the last couple of months, really powerful model systems that are derived from, of course, pluripotent stem cells. They can differentiate into both the epithelial and the mesenchymal lineages that are found in the intestine in vitro. But importantly, they lack really important cell types that you would find in vivo in the intestine, such as the enteric neurons, other endothelial cells, smooth muscle cells. So again, it's all about making these organoid models better, and making them better enhances their translational potential, of course, right? So here they demonstrated that the intestinal stem cell niche factor, epiregulin or EREG, can actually enhance HIO or human intestinal organoid differentiation so that they actually contain the epithelial cells, mesenchymal cells, enteral neuroglial populations, endothelial cells, smooth muscle in a single differentiation. So just this one hub protein, this one factor, epiregulin, has this dramatic impact on improving the differentiation of these human intestinal organoids and harboring all these auxiliary cell types. Importantly, without the need for co-culture. So we're not talking about like an assembloid system where you make one type of organoid and another type and you smash them together. We're talking about improving a single differentiation of these HIOs. And then they took it to the next level, of course, is a cell stem cell paper. So you got to do some of the translational applications too. They transplanted these HIOs into a mouse host, and they were able to further mature and actually demonstrate enteric nervous system function. And they had this like characteristic peristalsis that you see in the intestine. So they are able to undergo some of those peristaltic-like contractions, indicative of a functioning neuromuscular unit that you would actually see in the real intestine. And then the other part, as you might expect from introducing organoids with the vasculature into a host, is that the organoid vasculature actually anastomoses and connects with the host vasculature. So it forms functional vasculature demonstrated both in vivo and in vitro. So in vitro, they showed it using a microfluidic device, a microfluidic approach, and in vivo, of course, with the transplantation and the imaging. These HIO endothelial cells were again able to anastomose with the host vasculature. So that's really great. They're able to integrate, right? And so it's in big picture, it's a next generation tool. They call it a transformative tool, these human intestinal organoids for translational research in the human gut, potentially for drug screening, and also as importantly for disease modeling, being able to interrogate complex diseases of the gut, of the intestine, and testing therapeutic interventions with really high fidelity. It's all about making these model systems better. And I think the exciting thing about this paper is that this modulation of this one factor, epiregulin and ereg, that is able to extremely impressively improve the differentiation of these HIOs to harbor all these other cell types that you normally find in the human intestine. So taking these intestinal organoids to the next level.
Next level for sure. Transformative, I agree with that term because conceptually, I think it's not like a conceptual advance, something we never thought of before, but I think it validates or affirms something that we knew the whole while, which is that these things grow in place, right? They all these different cell types that are present in the tissue, apart from some exceptions where, they migrate to form multifocal sites of de novo organogenesis. For the most part, you get all these cell types that are growing out of whatever nidus sets them up, right? Whatever enlage. And so the fact that we're getting all these cells in there is super impressive to me, but it is really, I think, transformative in that it shows that you don't really have to take this more assembloid approach, which not that there's anything wrong with assembloids. I think that's more of an engineering approach, but this is, to the question we always put it out there to some of our guests, like, is it better to grow a thing or to make a thing? And of course, it's somewhere in between. I think here it's more the growth element. And there's a lot of cell types present in there, which is, I think, the focus of this story, why it's so impressive. But of course, I'm going to dwell a bit on the endothelial cells. The second to last author on this study, Shaheen, my old mentor, not old, he's still a young man, my postdoctoral mentor. And I was just visiting with him the other day because he's deployed this alongside of multiple studies. He's been on a streak. He just had a paper also in Science Advances that was very similar to this, where the vascularizing, not exactly similar in terms of the de novo growth, but using endothelial cells to vascularize pancreatic islets and showing their function was improved upon transplant, another transformative approach. But, to the whole angiocrine idea here, I would argue that the specificity of those endothelial cells growing out of that native enlage, I think, had a lot to do with all the other diversity of cell types, right? Because I have this endothelial bias, but I really do think it's real, this angiocrine facet of these organoids. And yeah, to multiply there on that, the anastomosis and integration with the host, thereby kind of utilizing both that kind of the vascular perfusion function, but also, I think, in the backdrop here, that paracrine function of the endothelial cells is really important to this story.
Angiocrine, Dalen, angiocrine. I mean, we just said that a million times. I know what this guy sounds like. I've never met him in person. So next time I'm in New York City, I gotta stop by.
You won't be disappointed.
No, I definitely won't. Yeah, this is a great study. I mean, we talked about the importance of the endothelium and actually driving the maturation of these things. Of course, this is near and dear to your heart. There's still definitely a lot of room for improvement here. It's a big step forward, especially just like I said, having this one molecule in ereg improving the overall differentiation of these HIOs in one shot. But there's still a lot missing here that just even modulating this one molecule can improve. And namely, I'm talking about the immune system. So you need to also have the immune system and lymphatic lineages, which of course play really important roles in intestinal function and disease. I don't know how you would improve that in an in vitro culture, perhaps some sort of immune co-culture. But in vivo, if you really want to take these things to the next level, you got to incorporate that immune component, right?
Yeah, absolutely. And as I said, I think the transformative element here is the principle of like the anti-assembloid, the growth. And I feel like it does introduce a whole other dimension of complexity to talk about, different heterogeneous vascular cells and different kind of functional vascular systems. But yeah, I think that this is something, again, that can be grown because that's how it happens, right? Physiologically and developmentally. So I see that in our near future, the way the pace of these studies is picking up. So yeah, you sent me your story about the liver, and I'm not going to lie, it was a bit thin out there with, strictly speaking, pluripotent stem cell stories, but it got me thinking of the liver. I learned a lot about the liver in my time with Shaheen. And it really is a tremendously regenerative organ, as we all know, but there's so much biology there that I think pertains to other organ systems, even though you wouldn't think of it being true because other organs aren't as regenerative. I think there are lessons that we can learn from the adult regenerative capacity of the liver and then apply and deploy them in our embryonic stem cell differentiation kind of systems. So that's my spin here on how I'm making this a stem cell story. But it's really about liver regeneration uh, and repair, right? And the cells that mediate that and the niche, because it's a complex niche that supports the metabolic function of the liver, right? You have mainly the sinusoidal endothelial cells, back to the endothelium, that, they're the main mediator here in terms of the perfusion, but also exchange, right? The influx, the efflux, exchange with circulation. Then you have the cholangiocytes, which are important for transporting bile acids and the metabolites that are excreted by hepatocytes and bringing those to the intestine. Then you have the Kupffer cells, which protect against the gut-derived microbiome and bacteria. And then you have these hepatic stellate cells, right, which are mostly known in like normal physiological healthy conditions for storing retinoids, and are the main driver of fibrosis in a pathological context. Like they're considered to be the central pathogenic cell type in the development of liver fibrosis. So that's where they get most of their attention. But they're the way they're situated, like where they are in the terms of the zonation of the liver, and their anatomy, like the way they look. These cells, they have a lot of protrusions. In terms of their molecular signaling, they have a really broad and expansive ligand receptor repertoire. And all these things mean that they interact, right? They interact with a lot of the cells in their neighborhood, including hepatocytes. But to this day, I mean, we know about them from fibrosis and the retinoid storage, but the function of hepatic stellate cells, let's call them HSCs, beyond that within the hepatic niche is not really well understood. So in this story, which was led by Robert Schwabe, who's at Columbia University, and also co-corresponding Helmut Augustine, who's at Heidelberg University, they looked at the role or the essential function of these HSCs, hepatic stellate cells, using a mouse model where they either genetically depleted them, and what they found when they did that is that they blew up Wnt, everything's Wnt, but they blew up Wnt activity when they looked just globally. But also there was like the zonation was all screwed up, and there were other alterations in the regenerative capacity, the metabolism, response to injury. And then looking, globally at sequencing, they found that RSPONN3, RSPO3, which is a Wnt signaling modulator, is dysregulated in this context. And then they go really surgical by showing that if you specifically delete this molecule RSPONN3 in the HSCs, they phenocopy the same effect of getting rid of all the HSCs altogether. which is to say the effect on hepatocyte gene expression. I think this is key and speaks to how these HSCs actually interact with hepatocytes, but also the anatomical defects in zonation, the size of the liver, the regenerative capacity, the metabolism, and also they get these mice on the sauce and show that the alcohol associated liver disease is exacerbated in the mice. And then taking it next, because this is a Nature paper, they actually show clinical relevance this in patients, showing that with, again, sequencing in these patients, that there's an inverse correlation with outcomes, showing specifically that there's a marked and progressive downregulation of RSPO3 RNA in the hepatic stellate cells of patients with advancing stages of fibrosis or alcohol-related liver disease or cirrhosis or hepatitis, right? So it looks like this is something that's really happening in patients. And here's another key that takes it back. I mean, in my own work, this is why it really jumped out at me, this story, because I'm looking at RSPO3 in my own, like it's a stem cell, pluripotent stem cell based niche kind of cell seed system. And RSPO3 seems to be a player in that system as well. Wnt, of course, modulation, not surprising, but specifically RSPO3 as a Wnt modulator. And it's not just me in this story, it's widely appreciated in a lot of other stromal niche and stem cell type organs, adult stem cell organs. So I think, like, if you wanted to apply this universally and specifically in our diffs, I think this idea of these modulatory niches here governed by RSPO3 of stroma can really have an important role in driving stem cell self-renewal and or differentiation, which is something we already knew, but I think there's a degree of specificity here that could be instructive for us stem cell biologists.
Wnt is life, Wnt is love. The answer to that age-old question of what is the meaning of life? It's not 42, as you learned in Hitchhiker's Guide to the Galaxy, but it is Wnt signaling and R-spondin 3 in particular. R-spondin 3, RSPO3, it's been, it's been studied for a while now. I think this is a cool angle, a cool tangent that they're able to put on this approach, looking at maybe the role of R-spondin 3 in regulating hepatocyte metabolism, regeneration, and so on. And I think there's a therapeutic and translational element to this story, as you're alluding to, maybe reverting these HSCs to quiescence or potentially even increasing R-spondin 3's levels could be like a therapeutic approach for liver diseases. But as we know with Wnt signaling, it's so ubiquitous, and it's all about the precise targeting of something as ubiquitous as R-spondin 3. So I think there's probably a bit more work to be done here.
Yes, for sure. And I think that it was really necessary for them to show that kind of descriptive link in a patient population to get this up to a Nature paper. But as you say, I think that there is in theory, while obviously more complicated and the proof of principle remains to be shown, I think there is this idea of therapeutic intervention. And for me, it's more about the idea, and this is again part of what I learned in Shaheen's lab, is how intimately intertwined the healing, the adaptive and maladaptive processes are in terms of like regeneration and fibrosis. And I think it really is modifiable in this case because you have a cell that's driving fibrosis, right? Well described as the major driver of fibrosis. And here we're finding that this is also the major driver of regeneration, but it's not on a cell intrinsic level, but on this kind of paracrine role with a specific target molecule. And as you said correctly, Wnt is everything, so you got to be very careful about your levels there. But I think that there's certainly something to move forward here in terms of RSPO3 as a therapeutic target.
Yeah, absolutely. And on the topic of regeneration and reversion, that's a nice segue into the next story that we'll be talking about. Another stem cell story coming from A friend of the show who's been on the show a couple years ago, a few years ago, Petka Kataisto. He's actually one of the first people that I interviewed when I joined the show, which is ages ago now. So this is a paper titled "Fetal-like Reversion in the Regenerating Intestine, Regulated by Mesenchymal Aspirin," and not aspirin as in Tylenol. This is asporin, Asporin. Just clarification there. We're talking about these kind of node genes, node proteins that are so important for a variety of cellular functions and influencing differentiation dynamics. We've already talked about two of them here on the show today, R-spondin 3, like the one that you just talked about. Here is asporin, an extracellular matrix bound proteoglycan, which has come up in actually previous Roundup papers. We've talked about it before. That was really the focal point of this particular study. So we know that mesenchymal cells, ECM, extracellular matrix, can support the epithelium during homeostasis regeneration across the body. And this is true across different tissue types. But the role of the mesenchyme in this process of plasticity and epithelial conversion into regeneration after damage isn't that's really not well established. We're trying to we've talked about this a lot on the show recently, right? This idea of cellular plasticity and how it's probably more likely than not that there's a conversion or reversion that happens in the body during times of injury, reverting back to a fetal-like state to perhaps kickstart those regenerative processes in the body, right? So what they did here in an intestinal model, they studied epithelial regeneration by culturing intestinal epithelium on decellularized small intestinal scaffolds. So this is a sort of a bioengineering approach that they did here. They call these intestinal epithelium scaffolds ECMs, and through their studies, they actually identified this asporin or ASPN as, like I said, an ECM-bound proteoglycan. And this is a critical mediator of this plasticity, this fetal-like reprogramming that happens. So, diving into a little bit more deeply, they saw that after damage, there's actually a transient increase in the expression of the ASPN, this asporin, by fibroblasts, these paracryptal fibroblasts. And these fibroblasts, in turn, induce TGF beta signaling, as you might expect, via CD44. And this is ultimately driving that epithelial reprogramming that leads to regeneration. So it's a lot of incremental steps here, but just to distill it down, this asporin ASPN is really driving the regenerative process here. And importantly, that's an extracellular matrix protein. It's a proteoglycan. So when you lose temporal control of ASPN in old mice, and they bring it to a mouse model, after damage, the high level of aspirin actually can reduce the level of regeneration. So it's stagnating the epithelia, is what they call it in the regenerative state. So that could be a contribution as to why older mice are not able to regenerate their intestinal epithelial as well. And bringing it back to the meaning of life, Wnt signaling, they show that an increase in Wnt signaling can actually resolve this stagnated epithelial regeneration program in the mice and promote restoration of proper tissue function. So back to Wnt signaling once again. And ultimately, they're establishing this platform for modeling epithelial injury, which you can use as an ex vivo approach to drive forward in vivo studies as well, showing that this one proteoglycan aspirin can modulate tissue repair and drive this plasticity going back and forth from fetal-like to adult-like state in a regenerative state. So here we are again. I thought we had gotten away from Wnt signaling, but we're back to it, Dale.
Yeah, I mean, didn't you just tell me that we can't therapeutically deploy Wnt because the levels are so critical? And here, there's just like, yeah, just put some Wnt, rub some Wnt
There,
on it.
there you go.
Um, I do. I mean, let's talk about the secret to life. Where can I get some Wnt, Arun?
Man, you can get it anywhere you want. Just don't get
I
it from
mean,
me.
shear? Do I have to take the shear? Where do I get the good shear? Anyway, the thing here to me, yes, I love, and it kind of fell out a bit. There was, I don't know, maybe it's like 20 years ago. They were talking about the decellularized scaffold. I don't know if you remember that, but they would like decellularize a whole pig heart or something crazy like that. And the scale was just too great. But that was, I think, more about approximating the anatomy and architecture, right? And then having the cells then fill. But since then, the ECM studies and decellularization is so much more about the instructive role of the ECM and the ECM bound proteins. So I love stories like this that bring it back. And I think it's a really underdeveloped field, right? I mean, we talk about the matrices that we're focused on here. It's Matrigel, I don't know, fibrin, laminin, about the most sophisticated, right? It's like the laminin 511, which incorporates the multiple subunits, right, to create something that has some kind of approximation, or like even the alginate with the modified epitopes. I don't want to say underdeveloped. A lot of people have thought about it, but I think in terms of recapitulating the biological, physiological presentation of these molecules, I don't think there's enough out there. So I like this. I think it's hard to maybe scale that up and make it commercial. I think it's going to have to be kind of bespoke at early stages. But as we move into standardizing, codifying these differentiation protocols, I feel like we're going to get to a place where it's not just about efficiency of the diff anymore, but it's about really getting these fine-tuned, like there's little things that we don't appreciate because we look at these markers that are, canon. But I think that there's subtle facets of the ECM input. And you know how you know that is if you take primary cells versus put them in vitro for one day, and everything changes, right? So there's something about the native ECM that's so key to the biology of every cell. And I really appreciate when it's highlighted in stories like this.
Yeah, as my guy Paul Burridge has told me, you gotta look under the hood and you gotta know what's exactly in your matrix. You gotta know what's in your media. You gotta distill everything down to its individual components, and that's how they're able to identify this, this one protein that's doing the thing here. This, aspirin. You're talking about decellularization. That was a really hot topic. I agree with you a few years ago. I remember when I was a grad student or undergrad, I learned about some of the work that Kit Parker was doing over at Harvard with decellularizing the pig heart. I think he was actually the one who's doing decellularization of the pig hearts. Um, and it was a really cool approach. You got rid of all the cardiomyocytes, all the cellular tissue. You're just left with this ghostly scaffold of the pig heart. And there was a translational element to this too. One thought was that you could just generate these decellularized pig hearts and then reintroduce the iPS derived cardiomyocytes and all the other cells of the heart back into the scaffold, and it would just kind of automatically and endogenously just reform the heart, just like a human heart with a pig scaffold. And obviously easier said than done. I think we've realized that that's probably not translationally viable, but it's just one of the coolest things that I had seen in my early days in science. And I think we've moved beyond it, but now there's other approaches for putting pig hearts into people. So, we're back to square one, I guess.
Well, I would say square a million at this point, the way science is advancing. You speak of artificial hearts. I just read about this titanium heart that went for 100 days.
Yeah.
We talk
Yeah.
about, these the use of the xeno. And so, yeah, I mean, progress marches on and everything maybe seems a little silly in retrospect. But the truth is we're still running up to the limitations of our capacity. And we need new technology to get there, right? And we are getting there in many ways, but in some ways we're still not getting the whole picture. And that's my segue to our final roundup story, which again, it's not even really a stem cell story, even less so. I don't think there's a stem cell in this story at all, but it's a method. I've been highlighting methods lately that I think are really applicable to stem cell science. And this is one where it's again about appreciating the native tissue environment so that we can duplicate it. I guess that's the theme that's kind of running through this episode of the Roundup. And I think that's a theme that's running through the ISSCR at this year's meeting as well. So maybe we could talk about that with our guests. But this is a tech story. It's from Nature Methods. It's about consolidating spatial transcriptomics and single cell sequencing, right? Because yeah, the environment is so key. We got the single cell, and we get really high resolution coverage of gene expression, but we don't see them in context, right? And that's because you can group the technologies apart from single cell. When you go to spatial, we're not quite there in terms of global coverage because there's two categories pretty much. There's a sequencing based approach, which has many examples. The ones that jumped out to me were because I've tried them, Visium and SlideSeq, for example. And then there's image based approaches, which are, I haven't used them, but I like stare at the pictures online because they're so amazingly beautiful, exemplified by like Merfish, for example, or Xenium, and maybe more commercially widespread. So these two approaches, one being the sequencing based and then the other being image based, are both fall a bit short, right? With the sequencing based approach, you have these predefined capture areas or the spots, right? And those can give you unbiased genome wide transcriptomic profile. But the problem there is that there's space in between those spots, right? These non-capture areas, as they're called. And depending on the resolution, which is getting better, apparently now the HD spatial platform Visium from 10x gets down to 2 micron. I haven't tried it yet. I was working with the 50 micron dots, and I have to tell you, it was not good. It didn't do the trick. And it wasn't just the space in between the spots there, but it was also that the spots were 50 microns. And I'm working in the ovary where you have cells that are like, I don't know, 5 microns. So you would get heterogeneity in that case. So it's hard to really get single cell resolution there. You get global coverage, but you lose coverage in terms of the whole space, and you get heterogeneity. When you go to the image based, which are very strong in terms of like the coverage, right? Single cell resolution. But with those, you're limited to a pre-selected panel, right? Around 300 to 1,000 targets, maybe more nowadays as the tech advances. But these approaches, I haven't used them, but from what I read, it seems like these approaches, the image-based spatial has, because it has limited counts per gene, they're better for like validation than for like discovery, and I know I stare at my single cell seq data and like really truly have epiphanies like, oh, that's that cell, right? Just imagine if you had the actual anatomy too. So it's a real unmet need, and this is a need that's been met now in this story by lead author corresponding Jiang Yu, as well as co-corresponding Junmin Peng, who both of those are at St. Jude's, and also co-corresponding is Kaibu Liu, who's at University of Wisconsin-Madison. And they dropped this new tech called Spotify, right? It's like Spotify, the music surface, except with a PH. That stands for Spot Imager with pseudo single cell resolution histology. Again, don't ask me how that lines up with that acronym, but it's good, it's catchy. And the tech is deep. Go look at the paper. It's more of like a bioinformatics type. It's not the way my brain works, so I just got the kind of the gist of it. But what it does, they showed with like really good benchmarking evaluations that you get this precise annotation of transcripts. And how it does it, by the way, I should backtrack a little. It generates what's called inferred single cell RNA expression profiles, right? So inferred scRNA-seq profiles, it uses generative modeling by incorporating 3 different data streams. One is just sequencing-based spatial transcriptomics, so like Visium with the spots, as well as just traditional single-cell RNA-seq, and then high-resolution histological imaging data. So with those 3 inputs, you're able to get this precise benchmarking, and they show that it's valuable in 2 use cases. One showing that astrocyte and disease-associated microglia, there's like these regional specifications in the Alzheimer's disease model versus healthy mouse brains. And then they look at human breast cancer data in context, cancer in the breast with the margins, and show that there's like multiple spatial domains and differences in like the tumor versus tumor microenvironment cells, like interactions. So, and that's like the takeaway from this with spatial generally, is that you get to see that niche interaction. And that's the takeaway from this whole roundup, right? What's the niche input? But I love this because I think that I was very disappointed, I'll say, with my experience with the sequencing-based spatial, and what I envisioned and what I got. It didn't really align, but I think that this, in just 5 years, has made amazing progress, and a lot of it on the bioinformatics end. And I think these technologies that kind of consolidate data in silico are going to give us really new visions of the tissue and the transcripts at a global level.
Spotify, huh? Yeah, I'm just hoping the lawyers aren't listening to this one. Just gotta keep an eye on those trademarks and copyrights. But really cool technology here. I mean, you're talking about improving resolution for all different types of tech. I mean, this isn't a stem cell story, but it's a piece of technology that can be applied, of course, to whatever you're doing in stem cell biology, whether it's looking at organoid sections or in vivo transplantation or whatever, right? You want to improve the resolution of your analysis. And I think that's just been a trend across the board for these technologies, this and related technologies. For the past decade, we talk about how single cell has become much more high fidelity. We talked about imaging improvements, two photon, and all these other approaches to resolution in your imaging. And this is looking at cellular resolution for transcriptomic analysis. So, in that vein, in that line, but still don't know about the name there.
Yes, when I back in the '90s, when you were like a fetus, uh, we used to say, that's not fat with an F, that's fat with a PH. I got to say, this tech is certainly it's got a PH in it, that's for sure. But I think you're right. They're gonna get hit with some trademark violations. But tech, as you alluded to, it's what drives progress. And a lot of the tech and discoveries are showcased at the ISSCR each year. And we're going to get a little sneak preview in our discussion with three heavy hitters on the ISSCR Planning Committee in just a few minutes. Before we get there, though, I have a brief message from Stem Cell Technologies. Do you want to implement the new ISSCR standards for human stem cell use in research? You can get a free checklist wall chart that helps you follow the recommended reporting practices and quickly reference the relevant sections of the standards. To learn more, visit stemcell.com/ISSCR-standards-wall chart. All right, everybody. Today we have what might be my favorite episode every year, because it's a sneak preview of my favorite meeting of the year, which is that annual meeting at the ISSCR, which this year is in Hong Kong. We're so excited. And we got the gang here, the leadership, to talk a bit about that meeting. First, we have Dr. Valentina Greco, old friend of the show, who is professor of genetics and co-chair of the Status of Women in Medicine at Yale University, also president of the ISSCR, if you didn't know. Also with us, we have Dr. Kathryn Cheah, who is the Emeritus Chair of Biochemistry and professor at Hong Kong University, also co-chair of the Program Committee of the ISSCR, and interestingly, chair of the Bid Committee that got the conference to Hong Kong this year. We were talking a bit about that before the show. Very fascinating. I didn't know there was a whole committee there and a pitch. But it makes sense now. It's obvious. But Hong Kong, it was an easy sell, I'll bet. Next, we got Dr. Eugenia Piddini, who is Professorial Research Fellow in Cell Biology and School Research Director at University of Bristol, also Fellow of the Academy of Medical Sciences. She's the co-chair of the program committee, also at the ISSCR. So that's the leadership that we're going to talk to today on the meeting. Thank you guys so much for joining us for this wide-ranging conversation.
Very thrilled to be here to share about the really exciting meeting that's going to be in Hong Kong this year.
And thank you for having me too. Welcome to the show, everybody. This is, like Dalen said, one of our favorite conversations to have every year, the pre-ISSCR annual meeting chat. We always hope this is a way to showcase the meeting that's happening in Hong Kong this year. And like Dalen mentioned, it's our favorite meeting of the year, the ISSCR annual meeting. It's happening June 11th to 14th. And of course, we're going to be there to recap the meeting as it happens, like we always are every single year. The theme for this meeting is, quote, illuminating the future, which is probably in part a reference to the city of lights that is Hong Kong. Just so to start things off, could each of you please give an overview of what you're most excited about for this year's annual meeting? Just kind of a brief snippet of what you're excited about. And Valentina, perhaps you'd like to go first.
Yeah, thank you so much. So I had the honor, privilege, and responsibility 2 years ago as I entered in this role to start to imagine what an annual program could look like. And so I started by figuring out what I knew and what I was ignorant of and deficient for. And that was a guide to figure out who would I partner with. So I'm clearly basic development, thinking about regeneration from a mammalian perspective. So I needed a lot of complementarity. So I started to speak with a couple of people, and one of them was Eugenia, to think whether I could find partners that were aligned with me, which were very curious about the things that we didn't know and we were deficient about. And I found in Eugenia an exceptional leader that was not only thinking along the same line of what complementarity can we think about the program, but they was seeking many more partners in order to do that. And so her idea became then to go around and ask with many colleagues, including Kathy, what were their vision and what were the possibility that a program could have in order to really match and satisfy a membership that is so diverse. And so Eugenia then identified Kathy, and then magic became.
Okay, so I was really blown away when Eugenia and Valentina asked me to join in to help shape the program. And I thought, oh, wow, this is enormous opportunity, not only to have a really good scientific program, but also to embrace their vision of making this meeting really international in terms of participation, the diversity of not only the speakers, but also the participants. And when I was making the pitch for the meeting to come to Hong Kong, one of the important issues I highlighted was that for an international society to be really international, it should represent as many of the researchers who are working on stem cells throughout the world. And in making the pitch, we made a pie chart. And we noticed in the pie chart that although, for example, in China, there was a Society for Stem Cell Biologists that had a membership of 1,500, whereas ISSCR's membership was 4,500. The number of mainland Chinese stem cell biologists who were members of the ISSCR was actually very, very small. It was something like 4%. And that meant that they were not engaging with the rest. There was untapped connections, not so much collaboration maybe, or anyhow, it was a very small representation. And so for the society to be truly international, we need to have many more people participating as possible. And so one of the important goals that we have is to bring in stem cell researchers from different parts of the world, not only from China, of course we have Japan, but then also from Korea, Singapore, Australia, India, very important. And for the first time in this meeting, we're having somebody from Africa, from South Africa. And so, I think for me, this is the most exciting part about this whole program. Not only is it scientifically outstanding, I hope that you will agree with me about that, but the breadth of participation is truly astounding.
Yeah, absolutely. Eugenia, what about you? What are you excited about, or what was your kind of focal point in assembling the meeting.
So I agree with everything that was said with how with Valentina, there was a true alignment with respect to the process and how we went about putting together a committee that would be as global as possible, so that it would allow us to capture an amplified network of stem cell community, so that we would be able to go well beyond their reach. And like Kathy said, the main motivation was that we wanted to build a program that was as diverse as possible, capturing diverse themes, diverse geographic representation, diverse career stages. And so to do that, we needed to bring on board as many people as possible that would be complementary to ourselves and to each other, so that we could, as I like to say, so that we could build a program that has both depth and breadth. And so I think this is what is exciting about this year's program. I'm very happy that working together with this amazing team, we had an incredible amount of fun. The committee is a lovely group of enthusiastic scientists who really came together and gave their best working with us. Valentina has been a wonderfully inspiring leader. Kathy has been my soulmate in putting this program together. We had an amazing chemistry and alignment of a vision, as well as complementarity. And I think this all together, I hope, will have really distilled the program that brings breadth of topics, depth of topics, and diversity of speakers in all shapes and forms.
Yeah, absolutely. I mean, I couldn't help but notice, looking at the program, how that jumped out at me. And it's amazing, Kathy, hearing that stat about how few of the massive group of scientists there, huge amount of work it's coming out of China now in our field, moving the field forward, and the fact that they're so poorly represented at the ISSCR is a real shame. And it's a loss, right, for us as a community. And, there's a risk of being left behind, at least in certain, parts of the field. But yeah, getting back to that global emphasis that you guys are talking about, it really jumped out at me. One thing I saw was this, what I thought was a new class of concurrent session called the Global Stakeholder Initiative. And as you guys just described so well, it's really about increasing the breadth, the depth, the diversity of science and speakers. But it's also kind of in terms of like stakeholder, it's a reflection of this pivotal phase we're in as the science moves into clinical practice, right? And the name is really appropriate because everyone at the meeting has a stake, but also everyone across the globe who is affected by degenerative disease and stands to gain from how we move forward in this next critical phase. But it's a bit risky, I guess, right? I think we've learned a lot of lessons from the past of maybe moving too fast or moving too carelessly. What is the overall idea with the Global Stakeholder Initiative sessions? Valentina, why don't you lead for that?
Yeah, I think so. In general, for us, was how do we take topics that are current, including we are wrestling with, and bringing them in a conversation. I wonder if Eugenia and Kathy can chime in for this in particular. But to me, one thing that is recurrent is not only in that session, but throughout, is how do we integrate conversation rather than separate topics? If I can just turn 1 second into an overarching view of the program, and then I'll let Eugenia and Kathy delve deeper, because this is really their doing, is how do we integrate a cross-session, different scale of thinking about problems, and integrating, including, for instance, health equity and DI perspective within session in the plenary times. So you will see a presidential symposium that aspire to go from basic development to tissue to disease-oriented, to implementation into clinic, to designing research in a way that all populations are served and not only some, integrated session. And so this idea, this ambition that Kathy, Eugenia, and the rest of the committee had is how do we create a conversation that is across scales avoiding it to fragment it and holding the tensions, for instance, from the implementation you were referring to, Dylan?
Yeah, so absolutely, like Valentina said, our goal is try to be as comprehensive as possible when putting together those themes that have inspired us to build the program, and to bring several perspectives. So I'll give you an example of how we have built in within plenaries a space for things that are beyond just science. And Valentina has already given you examples of that. Another one is a talk from Misao Fujita. She's professor of medical ethics and clinical psychology. And she at Kyoto University, and she'll discuss the costs to society and to healthcare of unproven stem cell therapies. So it's really an opportunity, while listening to Prime, the latest scientific innovation in stem cell research and medicine, also at the same time to reflect more broadly about the implications at all levels. In this specific case, as you mentioned earlier, there's a real push towards reaching clinical translation. And so this is really very much time to discuss, not just in concurrent or satellite sessions, but really within a plenary session where thousands of our community members will be listening about these important implications.
Mm.
So to add to that, I think like the ISSCR meeting last year was very inspiring because they had a special patient session where a patient shared about Parkinson's, the therapy that went with Parkinson's. And then we thought that, wow, it would be really great to let people see that where you all the way, as Valentina said, all the multiple scales that we go from the very basic to the translation. So we wanted to see whether we could also have a patient sharing session. And as it turns out here of course, there's a lot of logistics about bringing patients from other places, but it turns out that here in Hong Kong, we had a really good example of how gene therapy has actually impacted on a particular disorder, a very rare disorder, spinal atrophy, muscular atrophy, which is a very devastating disease. But it turns out, and we're going to be able to tell the story because we're going to have the clinician in charge of the two patients that we're going to bring out, who will share their experience. So the background goes as follows. The spinal muscular SMA is a terrible disease. It's, and it recently became a lot more treatable, or at least in terms of the symptoms, by gene therapy. But obviously it needs to be done at a particular time in life. So for children, it needs to be done early enough. But so there was one particular patient who had not had gene therapy, but had the other therapies which were not as effective. But she became a really strong advocate for wanting to make gene therapy, when it became available, to patients who are affected. But of course, it's very expensive. And through her advocacy, she touched the chief executive of Hong Kong, and she made it so that the government would pay for this. And so what we're going to have in this special plenary session with the patient sharing is the clinician who looks after the original patient, Josie, who was the advocate, and she will share her experience. And then the family of the young child who's been receiving the gene therapy and is actually really doing very well. So that's an example. I hope it will inspire all of us scientists who are working on basic mechanisms and models and cells and animals, that you can see at the end, if you make a contribution, it could translate itself to a therapy that will actually impact on a person and on people. And so this is like, I think, really inspiring that the society should make itself relevant to the public by showing this as an example, is that it's not all blue sky. And at the end, you can make an impact on somebody's health and their life.
Yeah, I absolutely love that. And I, like you, I love the session from last year that really drove home the importance of why so many of us are doing this work. It's for the patients, is to bring these treatments to people to help improve human health ultimately. So I think to have that framework, especially for junior scientists who are just starting to get into the field, I think is very important. And so on that topic of trainees, this is a highlight of the ISSCR. And as somebody who grew up on the ISSCR myself, I'm always so encouraged by the broader initiatives that are around that are at the annual meeting that facilitate trainees to attend the meeting. There's so many different networking sessions, meetup hubs, opportunity to meet editors from stem cell reports, discuss publishing. There's going to be career panels. So it's always been a great meeting for trainees. It's I'm always so appreciative of these initiatives. So could you talk a little bit more about some of these trainee centered initiatives at ISSCR this year, ways to support trainees financially to attend the meeting and otherwise as well? So Kathy, perhaps you'd like to start.
Yes. So let's say part of our bid was to raise money, right? And so when raising the money from the Hong Kong government for the ISSCR meeting in Hong Kong, we raised money not only to cover the cost of the venue, but also the registration fees of postgraduate students and postdocs and the early career scientists to cover their registration fee. And I'm very proud to say that the Hong Kong government did actually allocate funds for us to do this. Up to 400 will be able to attend if they would like to. And I think the last count is that up to now from Hong Kong, we have 150 students and postdocs who have registered because of the support of this free registration. This registration fee, is really essential for the ISSCR's health, but on the other hand, for many young people in especially developing countries, it is difficult to find the registration fee for them to come. So it's good that when they actually come, they have the opportunity to present, but we need to get them to come first, right? So I think this year is quite special in that there has been funding found to help the early career scientists to attend. And then hopefully some of them will be selected for an oral presentation through the abstracts that they have submitted.
Also, to build on Kathy, there is a membership international committee that is dedicated to think about abstract submission and decide for not only merit award, but also travel award. And this comes every year in large numbers. And there's been really an incredible capacity, not only to recognize and uplift, but also enable people to join. And so those are other mechanisms ISSCR has been using for a while to celebrate and bring in younger folks.
Exactly. So I happen to also be involved with the committee. And like Valentina said, it's an incredible work that the ISSCR does in recognizing the importance of lowering the activation barrier for travel, particularly for those who are coming from distant destinations and from low and middle income countries. And so the ISSCR has put in a system that tries to particularly support those whose geographical location and or country make it particularly expensive for them to join. I wanted to add, Aaron, going back to your question about activities that facilitate and celebrate our early career scientists, how much we as a committee, Kathy, Valentina, and I have enjoyed putting together the snapshot sessions. There will be, I think, 4 snapshot sessions this year. And these are essentially the topic and organization is led primarily by early career researchers. They submit an application to us. The snapshot sessions are about 90 minutes long. And they put together the program, the topics, they invite their own speakers. So it's a great leadership opportunity and really an opportunity for them to bring to the meeting interesting and complementary topics that have not been covered already by the bulk of the program, enriching the program with freshness.
Yeah, I love this emphasis, and we always try and highlight the trainees, but it's clear that I would say trending positively for years now, the ISSCR, but in particular this group, this meeting, it seems to be a major point of emphasis. And it's really gratifying for me because everybody remembers, everybody in the Zoom remembers, early going to your first meetings. And I go to the ISSCR every year because I know that the outcome is going to be so good. And it's such a huge part of science that everybody knows about, but you really got to experience and you really got to go after. And part of that is making it accessible, right? And part of that is really enriching the group of younger scientists who are coming up, and so they can really be highlighted. So I love that you guys are making such a big point of that. And yeah, part of that is because the fun of the meeting is like shooting around, right? Between all the concurrents. And I love that part of the meeting. But the plenaries obviously are it's a plenary because everybody comes together and not just because or not because there's any like difference in the quality of the science, but maybe because the content is of broader interest, right? Or broader relevance, or in this case, I think is very deliberately included to raise the profile of it so that maybe people who wouldn't otherwise be exposed to it because it wasn't a concurrent, we get them all. And that's what I've heard from you guys. And I saw one of them, the penultimate is inclusivity and equity in science and health. And Valentina, you've been a fierce, I mean, all of you, but Valentina in particular on this show and elsewhere, been a fierce, outspoken advocate along these lines. So I'm not surprised to see this point of emphasis during your tenure. But maybe you and the rest of the group here can elaborate a little bit more on, I mean, you've already said it. You got everybody. You want to get that in there while everybody's looking. But like in terms of like what the outcome that you hope for, right? What's like, you want to come away from this meeting and be like, we did it. What does that look like?
I can go back and think about, rather than what is on that stage, the process that brought to that. That's where I think we did it. The outcome may never be perfect. There's no perfection. There is learning and improving. But first of all, in order to get to what you will see in June in Hong Kong, there was a process that allowed for dissent, allowed for setting very clear lines of what are the values and the mission, and then mapping onto that any decision we were making and asking ourselves, are they aligned or are we deviating? Once again, trying to replicate ourselves, which is always a temptation, versus serving a community that is much broader and large, right? And so creating that accountability, but also creating spaces where the committee members could say, I'm feeling uncomfortable. We are running away from what we were saying, and us embracing it and saying yes. The colleague that now we learned from South Africa was because, again, Eugenia and Kathy were receptive to email receiving that said, I'm not represented. We are not represented. We have barrier. And they engage. And now these people have been also people that have engaged with other committee that we have created for ISSCR to rethink the awards. So the idea is, if you set the process where people are aligned with the values and you use it as a compass, then we can really create a system that will map it out and be much more matching of the membership we are serving. Second, the amount of work I've seen ISSCR team doing is unbelievable. You come into this conference like you, Dylan, I'm just excited to see people, but I don't think who puts up the light, the boards, the rooms, the coordination, the funding. Going to Hong Kong, while it's amazing that we had so much support, it means we are completely new territory and new uncharted funding system. So I've seen the ISSCR team essentially in bulk, just being oriented, sending lots of emails, creating new relationships. And the fact that it is international, it means you keep changing position, which means you don't have a stable relationship in that sense. So I think processes is my emphasis. Because by doing that, then we can say, okay, if this is the course and the turning that we want to do in the world that we dream and is not here yet, are we getting any close? And how do we go and keep ourselves accountable next year and the following year in order to get closer to that image?
How about you, Eugenia? What do you think would be the outcome that you would hope for?
Yeah, definitely. So it's a great question. I could give you so many answers, especially because we've talked so much about this program. For me, I guess the chief thing that would make me walk out of that meeting satisfied would be that everybody feels a feeling of inclusion. We have talked about the importance of having these discussions and raising our societal issues and how our community can be improved, including at the plenary panel, ADI panel. I think that level of inclusion and sense of inclusion, identity and engagement that we need to build within our society happens at that annual meeting when we walk into that meeting carrying our own little suitcase of experiments or research projects and plans and interests, and we carry that suitcase as we go around those seminars. And what we want to see is that our science is also represented and included in the ISSCR program and mission. And so that would be my biggest dream, that everybody walks out of the meeting having felt that their science was represented. We've tried really hard to do that. We've talked about how we have incorporated a number of topics that are not normally discussed within the plenary and concurrent, how we have also made space for patient and clinical translation and the ethical implications of that. But that we hope won't have come at the expense of celebrating the importance of basic sciences. We have talks that discuss not just mouse preclinical work, but also work that is done with genetic model organisms. We have talks that celebrate how Drosophila can be used to understand neural stem cells, or how intestinal parasites or planaria can be used to understand the properties of stem cells and regeneration. And that continues to be an integral part of what the ISSCR is about, really that breadth of going from the most basic science all the way to harnessing that for clinical translation. Our community at all levels allows this to happen, and maintaining that diversity and offering an opportunity for everybody to feel included in that mission is what I hope we will be able to accomplish with this program.
Kathy, I got a little wrinkle on that question for you, because you've made the sell. First of all, great answer, Eugenia. I just want to highlight how you guys are highlighting the right things, I think, and I love the way ISSCR is trending this year. But back to you, Kathy, because you've made the sell to the government and they bought it. What is it? What's in it for Hong Kong? Like, what is important to you about bringing the meeting to Hong Kong, for scientists in Hong Kong, for, I don't know, the economy? What is the real thrust of the sell there?
Well, in a way, we got lucky because the timing was right. Hong Kong is really wanting to try to develop itself as a hub for connecting East and West in terms of biotechnology and biomedical research. And the government is putting quite a lot of money into building new research institutes and also the mainland as well, to have this huge area called the Greater Bay Area. And I think what is in for Hong Kong is that because Hong Kong is such an accessible place, because Hong Kong has so many links with people, East and West, we hope that what will come out of this meeting is that the people will say, oh, wow, I learned so much. I learned, I discovered so much about what people are doing in different parts of the world in stem cell research, but also I've made enormous new friends, and I have increased my network tremendously. I know for a fact that the mainland Chinese scientists, I've been to the mainland actually to talk about the ISSCR meeting. And I've been so impressed and touched actually by the incredible enthusiasm. They're so proud that it's going to be in Hong Kong. They're so happy it's going to be in Hong Kong. And as a joke, one of the ladies who is a leader in the international relations for the stem cell researchers, Fan Yi, said to me, "Okay, Kathy, how many people do you want to come?" I said, "Oh, okay, 1,000, I said. She said, "Okay, 1,000. We'll see whether we get 1,000 or not." But anyway, I mean, but it's that level of enthusiasm. They want to participate. They want to meet other people, and they want to have their science heard, but they also want to hear the science from the other parts of the world. So I think come away, you asked what would I think would be the success. I think if at the end of this meeting, the people who attended said, "Oh, wow, that was such an inspiring meeting for us, and I made so many friends." I think that would be very satisfying for me.
Oh man, I can't wait. I mean, I think we can't forget that the first word in ISSCR is international, right? I think so many times we are USA inclined, especially, so many US scientists who think that stem cell biology or the best stem cell biology only happens in the States, but that's not at all true. This is an international society. There's incredible science happening across the world. Of course, that was highlighted in Hamburg last year. This year, it's going to be in Hong Kong, and we can't wait to meet scientists from all over the world, from mainland China, from Hong Kong, and just assemble, get together in this melting pot of culture that is Hong Kong. So one of the last questions I wanted to ask was actually also for you, Kathy, but as somebody who loves Cantonese food, I can't wait to get to Hong Kong just to experience the food. So I was hoping you could give some advice for first-time visitors about what to experience in Hong Kong outside the meetings. Of course, there's a food, dim sum, hot pot, the food stalls. I'm looking forward to trying some authentic sim chai key noodles, and all different types of food. So outside of the meeting, what should folks look forward to?
Actually, of course, Hong Kong is the food capital. I tell you, it is the food capital. And the really unique thing is that you can get food for a very, very small amount of money. And you can also dine in the best 5-star. So it's a huge a spectrum. And it's not only Cantonese food, because actually you will find represented in Hong Kong food from all the different parts of China, from Chiu food to Shanghainese food to Hunanese food to Sichuan food. But it's not only that, there's actually other kinds of food from Southeast Asia, Vietnamese, Thai, Indian, and of course, you can have fine dining, Italian and French. So, there's an enormous spectrum of food. And you can discover what it's like to have street food and go to the densest populated region in Hong Kong, in Mong Kok, and have, some strange parts of an animal. Dumplings, noodles, as you said. And you can go to the top restaurants, there are Michelin star restaurants as well. So, I mean, it's a huge diversity. But apart from food, Hong Kong has the most spectacular country parks and hiking trails. And this is something that, if after the meeting, people want to stay on and go and explore, there is incredibly beautiful country parks to go hiking and beaches to swim in. It'll be the time of year you can swim, so there are many things to see in Hong Kong.
Yeah.
Tourism board should hire me.
You did the job. You are making the sale. You're still in sell mode. Kathy, they already bought it. The meeting's happening. You are a very good advocate and spokesperson for both the meeting and Hong Kong itself. And yeah, as Arun said, I can't wait. we can't wait. We've been talking about this since, years, since it was decided. Looking forward to it. And I am going to be coming in hot because I'm flying in just the night before. So it's a good thing it's called the City of Lights, because I'm going to be a person of the night.
One last pitch. Actually, the day before the ISSCR starts, the three universities, Hong Kong U, Chinese U, and UST, we're having a pre-symposium in collaboration with ISSCR. And it's called Innovating the Future, the Power of Stem Cells for Medicine and Biotechnology. And what is meant, our target audience is high school students, undergraduate students, postgrad students. And we hope that this will actually inspire the young people to want to take up studies in biomedicals and especially stem cells in the future. So there's a one-day meeting where we will have speakers who are partly from the ISSCR, and Lauren Studer will be giving a talk, and Hong Kui Deng, who is not on the main program, but he's been in many programs in the past, and also is the recipient of the Future Science Prize for his reprogramming of cells. But also some of the local Hong Kong stem cell biologists and a few others from mainland who will participate. And the idea is to give for these people to explain in simple terms to the young people, to inspire them to want to study biomedical subjects, and especially maybe stem cells.
That's so good. I mean, these kids don't know. I say kids, the future. They don't know
the future.
What a big deal that is. Or maybe they do, probably, how intuitive and bright they are these days. But I mean, Deng and Studer groups have had quite a year and are at the forefront of moving our science into the clinic. And I think that's what this meeting is about, and that's what makes it so great. Is that you have the highest level scientists, in this case, speaking to the future and paving the way. So another highlight of the meeting. I'm glad you mentioned that. And I think I can speak for Arun. I mean, I'm born and raised in the city, but he's been everywhere. But I think I can speak for him in saying we both kind of look up to Hong Kong. So an amazing meeting, an amazing place. And I know it's going to be great. I'm really looking forward to.
So please come to the pre-symposium. Actually, I'll send you the information, and you should need to come earlier. That's on the 10th.
Will do. Right off the flight, Kathy. I'm rolling in hot. But yes, any other points that you guys want to make before we say goodbye? This has been a really great chat. Really enjoyed it, and looking forward to meeting all you guys in person. But any highlights?
Yeah, so there's one thing about the program that is different this year from other years that has not come up in the conversation, but that I think it would be important to highlight. So in past years, for those who are ISSCR annual meeting aficionados, they were used to finding a track on technologies. This year, our program committee decided to experiment with something different. And if you've looked at the program, you will have seen that we no longer have a track on technologies. And that was because a feedback was received from the community and the attendees that you're always interested in technologies, and there's so many concurrent sessions. And why would you have to choose between going to a technology talk when there's another talk exactly on your topic? And so we felt that perhaps to serve better our community and to, in fact, recognize that technology is embedded in everything we do, that we ought to incorporate technology within each track and just have technology-heavy tracks, if you like, where technologies are shown within the context where they may be relevant for your own research. So be on the lookout for that. You may not see technology highlighted per se as a track, but you will find it hopefully in every session.
Great.
Yeah, infiltrated. infiltrated throughout.
I guess I have one last comment I would like to make, actually 2 parts. When you were asking me, what do you hope the outcome to be, is that as people listen, they think and feel different ways in which competence manifests itself on the stage. And the definition of research is much more expanded than they initially came into the room thinking about. I think we have a narrow way to always celebrate the same names, the same people with the same identities. Arun was so wonderful in saying we often think US-centric, even though we are international. So we have already assigned a value to certain identity, geographic location, and so on. And so if people sit in that room and they think and feel that there are many different ways in which leadership competence contributions are made, from industry to ethics to health equity and so on, I think that will be an amazing outcome. Even if it is a feeling of dissonance, at least I think we'll have made a dent. And then also that people will come away understanding that this is not the doing of a single, 3, a committee, is the doing of so many. That conference is a mosaic of ideas incubated for a long time before us, ideas that came as a result of conversation we ever had, and ideas that came as a result of us getting it wrong until maybe we finally got into the right direction.
Yeah, absolutely. It takes a huge team to put on such a huge conference like this every single year, the biggest stem cell conference of the year, something that we're so looking forward to this year. So thank you so much to all of you for sharing your thoughts about this meeting. We can't wait to get to Hong Kong. The meeting's going to be June 11th to 14th, and we hope to see everybody there.
Yeah, look forward to that.
Thank you. See you there.
All right, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com. There you can get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X @StemCellPodcast or via email at info@stemcellpodcast.com with feedback or to suggest guests.